And I'm excited to be here. Thank you, Rashmi. Thank you for keeping it brief. Let's see if I can fail already on just sharing my screen. Let's see. Does that look like a real presentation?
Yep, it looks like PowerPoint to me.
And it looks like the audience view, not the
Is your presentation the slides? Yes.
Yeah, it looks good.
You look good. So I'm really glad to be here. And just my real phenotype is I have a PhD in computer science from the second heyday of artificial intelligence back in the 1980s. I'm trained as a pediatric endocrinologist, and I founded a department of biomedical informatics at Harvard Medical School, and I'm glad to tell you something that I believe may be interesting to this community. So it's going to sound like I'm talking about balancing a genotype first approach to complex disease like SGIA with a phenotype first approach, but really what it's about is enabling clinical trials to go faster and better and more effectively. And so over the next 15 minutes, I'm going to try to do the following. I'm going to try to convince you that broader and deeper automated phenotyping of SGIA using electronic health record data is technically feasible, because I've been doing it and others have been doing it for the last 20 years, and it's getting better and better and easier, and it can provide new biomedical and clinical insights. And it can be done across multiple leading sites of SGIA clinical care. And that there's a high payoff for the SGIA community, not least of which is regulatory approval and better trials. And I say this as someone who feels the urgency, whether it's in my own family members or the people I care for, around rare diseases, but really all diseases that are not well handled by the medical establishment. I'm privileged to be the principal investigator of the Undiagnosed Disease Networks Coordinating Center. This is a program that came out of the Undiagnosed Disease Program led by Bill Gall at NIH. And it's now a 12-site national program where we share all the data of our patients in the cloud, genomic and electronic health record data. And as you see from these statistics at the bottom, we've seen thousands of patients and really made a difference, a meaningful difference in diagnosis, probably for somewhere between 25 and 30%. So that's my way of persuading you that maybe I have something to say here. It's important to start by saying, I'm going to use autism as an example, and then we'll put it to SGIA, to say that the genome first approach, where just looking at cases and controls, autism versus non-autism, has been incredibly successful. And in works supported by the Simons Foundation, for example, we found hundreds of genetic loci that were implicated in both rare and common variants contributing to the risk of autism. Not the full story, probably somewhere about 25 to 30% of the inherited risk. But let me give you a story. I'm pediatric endocrinologist, not a neurologist, but I was invited by a mom to get more involved. I did. And when I started studying autism, mothers came to me saying, what's with our bowel disorders in these kids? And I did a large multi-site study across the United States of using electronic health records. And I found that there was something like up to 11% incidence of bowel problems, including inflammatory bowel disease in autism. When I presented this both to GI doctors and developmental specialists, they were skeptical. And so when I went to one of the leading ones, and he said to me, and it was a little bit dismissive, brain bad, tummy hurt. I kid you not. So I went to this then postdoc of mine, Finali Doshi, who's now a tenured professor of computer science at the School of Engineering and Applied Sciences at Harvard. And what she did was very simply, she took the first 15 years of these patients, of each of these patients through the electronic health record. And I'm going to spare you the details out of time and because you might find it boring. We clustered the patients, and we clustered the patients so that there are groups of patients together who had similar comorbidities, not psychiatric, not necessarily behavioral abnormalities, but through the full medical record had similarities in the same periods in development. And what we found was not one autism, but multiple autisms. This is the network we use across the United States. We found one subgroup absolutely loaded in seizures, one 80% seizure, another distinct subgroup with a lot of infectious disorders, both viral and bacterial, and also autoimmune disorders like inflammatory bowel disease, and yet another group that had increased load of psychiatric diseases, including schizophrenia, ADHD, and anxiety and depression. And we've done this approach using electronic health records to dissect the subphenotype of these patients for many diseases, squishy diseases like major depressive disorder, less squishy like inflammatory bowel disease, medium squishy like post-acute COVID. It's doable, it's just work. At this point, it's not rocket science. I will just quickly, so we've done that, sorry. Those are the three subgroups. Just to tell you that we can then take those phenotypic insights and then get some better molecular insights. And so, for example, to make a long story short, we looked at all the genes in patients in autism who, where the genes during brain development were very different from those in males versus females, because autism is 4:1 male-female. And using that phenotypic insight, we were able to drill down and find those genes, and of those genes, found which rare mutations of those genes travel with the patient phenotype. And we found several pathways, but the novel pathway we found was in lipid regulation, basically the metabolism of fats. And when we looked at now at this pathway, lipid disorders in electronic health records, we found that at Children's Hospital, there was way overrepresentation of lipid disorders in the kids. And that furthermore, we found the same thing in the claims database, and more importantly, in the kids in the database who were siblings of the affected kids, they did not have the same dyslipidemia. And so this led to a Nature Medicine paper, but the point is we were able to use the phenotype and the genotype together to get better insight. So I'm going to try to set the stage by keeping on time. So here's my suggestion for increasing the reach and precision and the precision medicine analysis of SGIA. I'm going to say that SGIA, like all complex phenotypes, even rare disease complex phenotypes, is not one disease, but it's a multitude of diseases. And I'm guessing, without knowing anything, that, well, actually, I know a bit because of Rashmi, that there are many subphenotypes in SGIA, and they're not well recognized by the medical establishment, just like the bowel disorders were not recognized by the autism establishment. I can't tell you how many thankful moms I had come to me in tears of gratitude, more than anything I'd ever done clinically, because I was recognizing something that the medical establishment had not realized, and we're treating, and they could therefore start treatments for something that was otherwise pretty terrible. So here's the important deliverable, and I'm really trying to get concrete rather than show off. I just want to give my best possible advice. I think we should have, there's an important deliverable that is actually deliverable, which is natural histories of our SGIA patients without bias of the different types, all of them, for regulatory approval and evaluation of small single-arm trials. I can tell you now, having worked in this rare disease space, FDA, there are very well-funded companies like Ultragenyx who've gotten FDA approval based basically on comparison diaries to see the progression of disease. We can do a lot better than that by going across the major health establishments, taking care of SGIA kids, using the electronic health record to find the trajectory, just as I tried to show you that we were able to do around for autism. And so by creating, here's the term of the art, computable phenotype that can be run against clinical nodes, that's important, and the codified data. So both the labs and diagnosis, but also what's in the notes, because we found that to be much more reproducible than some of the billing codes. Make it open source and portable. So you're making it for the world. And you can decide, so here's where my PhD in computer science is unfortunately showing its nasty side. There's two ways you can do this. One is supervised versus unsupervised. In plain English, you can just say, here's everybody, find me the clusters within it, and in a completely unbiased fashion. Or we really think that clinicians know what they're talking about when they look at, let's say, the lung phenotype, and we're going to stratify around that. So either to let the computer decide for itself what are the natural subclusters of this disease complex, or you give it some hints using a supervised method. Long discussion about which way might go better. I'm getting very concrete here. I would pick 2 to 3 major clinical sites for SGI care. And I know they're well motivated. It'll get them papers, they'll get published. Everything's aligned, but most importantly, it'll give you the tools to actually show the drug companies and the FDA that you can actually have the natural history to compare it to these patients. And by the way, all of a sudden you're puffing yourself up much bigger because you're not just this and that study. You're coming together community, and it doesn't have to be big. I'm a big fan of perfect is the enemy of good. Don't do this. Don't get everybody involved. Just get the few sites that are ready to move today who are willing. And then you can do the kind of stuff that I briefly rushed over in the Nature Medicine paper, which is once you have the phenotypes, you can go and look at a particular subphenotype as defined by the EHR of these patients. And we've done this, by the way, for seizures. And really, it works again and again. And then you can do the expensive studies and looking at these sub-diseases using single cell sequencing, using whole genome sequencing. But first, get down in an unbiased way. What are these subsets and what are their natural history? If you want to see more of my screed about this, I put something out in Neuron a while last year with my perspective of this. And last of all, I want to bring it back to where it starts. Yay, moms. And I say this not in a paternalist way because I've done this with my own family. There is no one else who feels the urgency of the suffering of our loved ones more, and I say this as a doctor, than patients and the patient families. You guys, those of you who are parents, there's only you in order to be the forcing function to get this done. And it's doable. End of story. I'm done. Rashmi, sorry for going along and sorry for speaking so fast. I'll blame my tooth.
No, that's great. I think we have time for a few questions. So I'm going to make sure that does anybody have a question that they want to speak up?
And by the way, Rashmi, I want to point out that I went fast because you told me that yesterday you went long into the break. And since I host many conferences.
No, I think thank you for that. I think you've set up the stage really well. I think it would be good to have a little bit of discussion. As you know, we have an ICD-9 code. It is a rare disease. Some of your examples were from larger diseases. So like, what data sets would you suggest in looking for SGIA? Like the specifically?
So let me quickly ask you.
Yeah.
How many patients do you think there are in the United States with SGIA?
In our group, we have about 1,600 parents. Leah and I are best guesses, but Hermina, etc., might have a better idea.
Well, that's good enough. That's
good enough. Okay.
Order of low thousands, and
okay.
So let me tell you, that's not that rare. And so, for example, I have a slide I didn't show you around tuberous sclerosis, where we did this using claims data across the entire United States. And we were able to create the largest tuberous sclerosis phenotype. And we found subgroups of tuberous sclerosis. We found some disease, some comorbidities they didn't know about. We found some groups with some cancers that they didn't know about, and we found actually protection against certain cancers. So I think so to answer your questions, Rashmi, if you were to, so what are the databases? I think there's two classes of databases. And if I were you, and with all your energy, this is what I would do. I would say, pick the three largest. So I need to do this in top-down fashion. On the one side, pick the three largest clinical sites and become really friends with the CEO of United Healthcare. And so the CEO of United Healthcare is the CEO of the largest private insurer in the United States. And they have all the claims data of patients. And you cannot run away in the United States. You can actually leave Cincinnati Children's or Children's Boston, and you'll have an incomplete record because you're being seen elsewhere, but you cannot run away from your bills. And so the claims data has most of the history, plus it has family relationships, unlike the electronic health record. The electronic health record, the clinical data, you can create a data mart, fairly straightforward. It's a very simple conversation. It's just politics with the CIO and leadership about creating an extracted data mart that's clinical notes, the labs, the medications, diagnoses, and demographics of the patients with SJIA or seen in an SJIA clinic. And so that's the three sites. Then you talk to the CIO, Larry Renfro, great guy, head of United, and you say, I want to do this study. We'd like to look at all your patients. It's going to be anonymous, as opposed to, and so the first one, the EHR, is for the trajectories, and the work with United is going to give you a couple things. It's going to give you some trajectory, more coarse-grained. It's going to give you the dollar cost of each one of these patients. And it's going to give you a much more unbiased sample at the nationwide level.
Okay, I think I grabbed that, and I think the focus right now is to find the refractory patients. And give me the example that you'd mentioned to me earlier, where for some disease you defined it as refractory. Yeah.
So major depressive disorder, squishy disease, and we wanted to look at the genetic signature of major depression resistant to standard pharmacological treatment. So what we did is we looked at those who changed drugs, medication once. That's a smaller group. Changed drugs twice, another even small group. Changed drugs thrice, even small group. Actually, unfortunately, because major depressive drugs don't work that well. Turns out actually it's 2/3 of the people change drugs 3 times. And then because we really wanted a strong genetic signal, we said, and those who go on to electroconvulsive therapy. And that intersection gave us the most refractory patients with major depressive disorder. Similarly for refractory epilepsy, and we're working with a drug company about this. We just identified those patients who've gone through the most drugs. They keep on changing drugs. Why? Because they keep on, they don't respond. And you can see admissions, emergency room admissions for out of control care. I can see exactly how this is mappable, broadly speaking.
That makes sense. That makes sense. I think there'll be a lot of medicines, and like the number of admissions and the number of ER visits, etc. You could concretize it like that. I think we have a comment from Hermina in the chat about these that the data mart at Cincinnati Children's. About the kind of data that might be worth looking at.
Just to add on what you said, there's lots, there are PedsNet data. Most of the pediatric hospitals in the US are on Epic. And actually the large pediatric hospital has worked together to merge the Epic data. So that's another area where you can get good data. There's also some commercial entities like the FIS database, which you already provided, and also the TriNetX, which gives you actually global data. So there's a lot of data out.
There are. So you're going to have to take this from me on faith. So TriNetX, that company, actually built the technology over the open source I2B2 technology that we developed. I'm very familiar with this ecosystem, but I'm trying to give you, so PedsNet is great, but what PedsNet is not doing yet is using the clinical notes. And that's why I'm focusing on the subgroup of hospitals, because we found that for squishy diseases, for example, rheumatoid arthritis, we found that fully half of the patients who were diagnosed with rheumatoid arthritis at the Brigham did not have rheumatoid arthritis. It's just because someone ruled out rheumatoid arthritis on the procedure, on the radiology procedure, so they could get paid. But then when you automatically read the clinical notes, they were ruled out. So I do think that if you want to have an accurate FDA-proof clinical trajectory, you're going to need to do natural language processing on the clinical notes. That's my best advice.
I agree, especially in the current time where if one want to have a medication from an insurer, you have to look at the diagnosis very carefully, and you may have to morph it to decrease the pain for the appeals and so on.
Yeah. So there's a lot of black arts around this, but it's doable. It's just work. It really is just work. And we did several disease projects where we just got together with leaders, worked it out, and since people at Cincinnati Children's, for example, are very good at this as well.
Okay, any other different questions to bring up, and then otherwise we'll move on. Thank you so much, Zach. I'm sure we'll follow up about this. This is
Regardless,
think differently.
Yes, you really do need to think differently because, again, I've seen the patient groups have succeeded is you don't wait for the medical establishment. You've got to bring the resources together now, today. And the good news, it's easier today than it was 10 years ago. So I strongly, and you have collaborators. You actually don't need me, but you have great collaborators locally and internationally. So great seeing you all. And I hope one day we get to meet in person.
Yes, thank you. Thanks.